Yeah. So cool. Georgi, can you do something? Like, do you have any access now? You co-host? Oh, yeah. Good. So, I think we can start. If somebody join, um, somebody will join. So let's go.
Okay, so I will start with the basics of learning process with such mechanisms as habituation and sensitization. And let's begin. So, wait a second. Yeah. What's the origin of the whole problem or study of these processes and the whole field of memory and learning neuroscience? So, initially, these studies were conducted by Sir Charles Sherrington. I think he was one of the first neuroscientists who performed experiments usually on cats. And the problem with cats or some other high order vertebrates was very complex brain structure. So, for example, if you consider even just one brain region, such as hippocampus, you know that there are a lot of cells there that have different types that they wire between each other in a very intricate manner. So, to dissect such complex cognitive functions as learning and memory, it took a lot of effort, and even these results that were obtained during these experiments were very confusing. So, very few progress was made until the beginning of Eric Kandel work. So Eric Kandel was a neuroscientist that was born in Vienna in 1929, and initially his interest lied in the field of psychoanalysis and psychology, and his training was in these fields. But after some time, he realized that the approach of psychoanalysis, when you study the brain as a black box and you do not know which mechanism, which circuitry you have in the brain when you try to explain phenomena that psychoanalysis deal with, he decided to switch to biological approach. And that's why he spent several years studying medicine and the USA. And as he was always fascinated by such problems like memory, learning, he decided to do electrophysiological research. And the main problem that I already described before, with the complexity of the brains of the higher order animals, was also the same for him. And of course, he needed to find an object that would be very helpful in his research. And after some time, after some search, he stopped his uh, attention on Aplysia californica. So, what is Aplysia californica? It's a very common sea slug, so-called sea hare, because some tentacles that it has on his head uh, remind people of the ears of the hare. And the whole um, journey of Eric Kandel ended with the Nobel Prize in 2000 uh, in physiology and medicine. And now, just small history remark. Now, later work of this famous scientist was dedicated to the merging of neuroscience and psychology and psychoanalysis. And he later proposed so-called neuropsychoanalysis. So the idea was to merge anatomical and molecular findings that happened during the rich history of neuroscience and findings that were made by psychoanalysis. So the idea was to put a biological background on phenomena that are usually studied in psychology and psychoanalysis. Uh, as far as I was at some point very interested in the research of this famous scientist, I read some of his books and some of his papers, and I just wanted here to make a small step aside to recommend you these books that he wrote because he was not only a great neuroscientist but also a great psychologist and psychiatrist. And I think he was also, because he was born in Vienna, one of the most uh, famous cities, like where the different cultural phenomena, different art directions aggregate, so he also was quite um, educated in this field, and he also tried to explain some of our preferences, for example, in art, some of the motifs that we see in art by biology and psychology, by means of biology and psychology. So I highly recommend these four books of him. Maybe you would find them interesting. But let's switch again to Aplysias. So why we talk about this specific, very strange object, not common in neuroscience at all? I mean, before the findings of Eric Kandel. So you can see here the photo of this sea hare. It's quite big, so I think it's the has the size approximately of a small hare or rabbit actually. And here you see the list of the advantages of this model for the neuroscientific research, especially for the learning of memory, for the study of memory and learning. So here on the right side of the top, you can see the ganglia of these mollusks. So these mollusks are very primitive invertebrates, and of course they do not have something like humans or other vertebrates. They do not have a central nervous system. They do not have a intricate brain structure, and all the neurons aggregated in these ganglia that you can see here. So just one of them. And these ganglia also connected between each other to form this called, I think it's called ladder nervous system. And usually each pair of ganglia are responsible for some specific functions. For example, pedal ganglia controls the movement of the leg, and cerebral ganglia is the main processing region, the region where all information flow accumulates and relays between other ganglia. Uh, as you can see here, the neurons are quite big and very nicely distinguishable. They're also colored. It's a common feature, I think, of all these branch of mollusks that the ganglia and the neurons itself, they have this very bright orange color that you can easily find them when you dissect the animal or when you record them with a patch clamp or microelectrodes. Actually, you can see it here. And another big uh, advantage of these neurons of these animals is that they are very big. So, for example, the biggest neurons could reach the size of 1 millimeter, and you can actually see them by eye without any microscope or binocular. Another important thing is that, as you can see here on the bottom on the right, each neuron could be identified, and all these identified neurons are unique. So, because these mollusks has a special determined type of development, each uh, representative of the species has the same neuron at the same position, and exactly the same neuron will be responsible for the same function in all the animals of the same species. For example, L2 neuron could control some specific movement of some specific muscle, and some other neuron could be a pacemaker, for example. Some other neuron could be an important interneuron that would actually govern sensitization or habituation that we discuss later in details. And because these neurons are so easily identifiable and could be mapped on these specific maps of the ganglia, you can also extract them and use them not only for electrophysiological recordings, but for example, for molecular and genetic analysis to see the RNA expression or do some other sequencing techniques, perform some other sequencing techniques for any means that you want to do. And what is also really important, because we remember about cats or some other animals, that the problem of the behaviors, of complex behaviors and complex cognitive functions, is that they're governed by a lot of hundreds, thousands of neurons. Here, some actually very important for the survival of these mollusc behaviors, like gill withdrawal reflex. We will talk about this in the next slides. Usually governed by two neurons, so one sensory neuron or one motor neuron. And then, for example, if we talk about learning, some other neurons could be attached to the process, but usually the simple neural circuit, the simple chain, is two neurons and some effector and afferent organs. So just a little bit about the anatomy that would be important for the discussion, because it's the mollusk that lives in the on the bottom of the seas, they have a siphon by which they excrete uh, some food, like food residues, and the water, and the gills that are the organs of breathing for them. And the gill and the siphon, usually in these species, are covered with a big folding, so-called mantle. And one of the important types of behavior of these mollusks is the withdrawal of the gill and the covering of the gill and the siphon by the mantle. So to protect it from damage by rocks and, for example, to prevent it from damage that can be caused uh, by the animals that hunt them. Sorry. So, and let's talk a little bit in details about this so-called guilt withdrawal reflex. So you can see here that the siphon and the gill are connected, and if you would squirt the water in experimental conditions in the tank with these animals on the siphon, you would see that the gill actually contracts and withdraws. And if you would continue squirting the water on the siphon with a constant flow of the water, you would see that actually the contraction is stopped. And if you would measure these for several minutes, sorry, then you would see that the power of this contraction decreases and then reaches some plateau. And this is very interesting because here we see that the animal learns that this squirt of water does not provide any damage or danger. And you can see that here we see some process of learning, very simple one. So that was initial idea to study the neural mechanism, molecular mechanism of this learning process because it's very simple and you can reliably detect it by behavioral experiments. The anatomy that of the whole reflex that was revealed by Kandel and colleagues is like this. So here on the picture, you can see the head ganglia and abdominal ganglia, which are connected by neural commissures. And as I said, you can see the two neurons that are involved in the process of this gill withdrawal. The sensory neuron that is connected to the siphon that senses the flow of the water. These sensory neuron processes, like the body of the sensory neuron actually lies in the abdominal ganglia, and then it connects to the motor neuron that controls the contraction of the gill. And the simplified circuitry you can see here, so the sensory neuron, the motor neuron, and the effector and efferent organs. So as I told you already, this is a very simple system, very simple model. And the second question that appeared, how actually the process of this habituation, this attenuation of the reaction of the gill occurs. And even though there are only four members of the circuit, so two organs and two neurons, habituation possibly could occur in different regions. So it could be occur somewhere in the sensory neuron, or in the on the level of connection between motor neuron and the gill, or for example between sensory and motor neuron in the region of where they connect between each other, these synapses. So experiments with this water stimulating and recording um, the responses in different neurons and also in muscle in the gill were performed. And as you can see here, if you record from sensory neuron while stimulating the siphon, or when you record from the muscle while stimulating the motor neuron, you do not see any changes over time in the responses of the muscle, so it's the muscle tones, or in the action potentials in the sensory neuron. So what have Kandel and colleagues found that actually attenuation occurs in the synapse between sensory and motor neuron, because you can see here that the excitatory postsynaptic potentials, sorry, so that you can record in the motor neuron after stimulating it by after sensory neuron stimulates it, you can see that these responses become lower and lower over time. So that was the first step to explain the whole mechanism. But the second step would be to explore to explain it on the molecular level. So like what actually happens in the synapses. And if you remember, the action potential that flows to the axon terminal in the sensory neuron produces the opening of the calcium channels that insert the axon terminal, and by some different molecular processes govern the excitation, or not excitation, sorry, the release of neurotransmitters that connects to the receptors to these neurotransmitters on the motor neuron, and these generates the extra excitatory postsynaptic potential. So what happens if you would stimulate, for example, the gill, or if you would touch the skin of the cat, doesn't matter because this process is very fundamental for all the animals. Or for example, if you would continuously stimulate, I don't know, by your voice, your um, friend during some discussion. So in some neurons that are stimulated, you would see that the amount of these vesicles that carry the neurotransmitters would become lower. And the number of these vesicles that are connected to so-called active zones right on the on the surface of the membrane that is close to the synaptic cleft, these numbers would also become low. And here we see two different types of these habitations. Short term, as I described already, so just lower amount of vesicles and lower amount of vesicles in the active zone. And short term habitation that could last for like hours or even weeks or sometimes months. Here, beside from the lack of the vesicles, you can also see that the action zone size reduces over time, and you can see this by performing, for example, electron microscopy slices and imaging of these synapses. And again, as well as in the short-term habitation, reduced number of vesicles attached to the active zones occurs, and reduced number of the vesicles itself in the axon terminal. So these are the molecular and circuitry mechanisms of habitation. And this was the first big part of the research of Kandel. Another process of learning that we occur in all the animals, very basic and very simple, is sensitization. When we talk about sensitization, we can think of, for example, you listening to the lecturer, and then you become bored and you fall asleep, and then something happens. I don't know. For example, you see the fire alarm, or just the teacher claps his hands, and you become very alert and attentive. So after the process of sensitization, you would see that even the stimuli that were subthreshold that never draw your attention to them, for example, I don't know, some sound of falling pen, they would actually trigger a very sharp response. So you would actually be very triggered and nervous, for example, if we talk about you. And what happens in aplysia is sensitization of the kill response. So, for example, how it could be achieved, this sensitization on the behavioral level. You can apply electric shock or some other very irritating stimulus to the other part of the body of the aplysia, for example, to the head. And then you would see that even though nothing changed in your treatment of siphon or the kill, you would see that the neurons that trigger the activity of the gill, this gill withdrawal reflex, they become more active, and the power of contraction of the gill actually um, stays the same during over time, even though you never triggered this reflex on its own. What has been found by Kandel and colleagues, that the location in terms of neurons is the same as for the location for habituation. So again, this learning process happens between sensory and motor neuron. But what is different here is the presence of the third player, the interneurons. So how the whole circuitry of these reflex, these and these in regards of this novel mechanism of learning works. We here have the same connection. So the sensory neuron is connected to the siphon, motor neuron is connected to the hill, and the synapse between them. But we also have the sensory neuron, for example, in the head. And you remember that we perform these electric shock, for example, on the head. And this sensory neuron senses this irritating stimulus and transfers the information to the interneuron in one of the ganglia. And what is interesting here that these interneurons, they call it usually L29 and L28. It's a specific neuron dedicated for these specific learning mechanisms in these mollusks. They have their axons connected to the axon terminal of the sensory neuron, and they can modulate the activity of the sensory neuron uh, regarding this reflex. So what happens during this shock sensitization is the increasing of the power of the, let's say, of the excitatory activity of the sensory neuron axon terminal, and the excitatory postsynaptic potentials that are produced by the released neurotransmitters from the sensory neuron in motor neuron become much stronger. So this leads, of course, to the stronger response of the gill, stronger contraction. And I think I would not dive in details here in all the molecular details because I think that maybe also Zhenya would discuss it with you on in regard of mice experiments with mice. But uh, what what are the main mechanisms happening that allows this sensitization to occur is the opening of the calcium channels, increased time of opening of these channels that trigger the uh, excretion of these vesicles with neurotransmitters, uh, increased production of these neurotransmitters inside the axon terminal, and also increased, let's say, merging of these vesicles to the axon terminal, so that which also in turn allows you to produce to excrete more neurotransmitters in the synaptic cleft. And as well as for habituation, this process could be short-term and long-term. If we talk about long-term effects, it involves some changes in the regulation of biochemistry of the cell and of the gene regulation in the cell. And you can see the summary of the whole cascade here. So you can see actually different mechanisms that in like all together allow this sensitization to occur. So, for example, the incorporation of a new calcium channels, incorporation of a new potassium channels, which allow the by three different mechanisms also to release more neurotransmitters and some genetic changes that occur in the cell of the sensory neuron. So here you can see that these findings actually helped us to understand the behavioral mechanisms, the circuitry mechanisms, and molecular mechanisms of these very simple forms of learning that are interesting for us, not just as a fundamental thing, but also because all these mechanisms are conserved across species. Of course, we can find some differences in terms of some gene regulatory networks, for example, in different phyla of the animal kingdom. But still, these mechanisms underlie the all other more advanced learning um, mechanisms, for example, in humans or in other birds. And this was very essential finding. So actually, that's why it was awarded by the Nobel with the Nobel Prize. And we talked with Eva a little bit about this. So as you can see, if you would read the books from Kandel, for example, you would see that the whole story of this finding is a very, it looks like you can read it like as a detective or some kind of adventure roman or something like this. But one of the things why it actually happens is because the object of the study, as you can see, is very unusual. So aplysia was something very novel for neuroscience and I think for biology or electrophysiology, physiology in general. And I just wanted to talk a little bit about the importance of this comparative approach and the importance of the good selection of the model organism for your research. It doesn't matter if you study learning, memory, or vocal communication, I don't know, synapse formation, plasticity, or something else. So I think more or less at the same time, experiments on other specific mollusks were happening. You can see them here. So this is a very unique mollusk called sea angel. And these sea angels live only in the northern seas. Usually they could be found, I think, in the North Sea and in the White Sea. And here we talk a little bit about the story of the comparative studies in my country. So here you can see the two main participants of this, main players of this story, the father, let's say, of the comparative physiology and comparative, more important, comparative neuroscience in Russia. So it's Khachatur Kostoyants. And his descendant, his student, Dmitry Sakharov. And what has been found when this mollusk has been studied, it was a very unpredicted finding because nobody was never interested in them as a source of material for neuroscientific or physiological research. What has been found is that the neurons of these mollusks are more or less the same as the neurons of Aplysia, so they're also pretty big, a little bit smaller, but still. They have the same program of development, these determinate development, where you can see the specific neurons located in specific positions, and you can map them and identify them very easily from animal to animal. And the recording of these neurons were actually really easy. But what's also interesting here, so if you talk about aplysia, you see that behavioral patterns that it has are very simple. So, for example, guilt withdrawal reflex is very basic. It's only two organs and two neurons, or maybe some interneuron if we talk about learning. But here it's a little bit different story. Here you can see that if you would observe the behavior of these mollusks, that they are very uh, good hunters. And you can see that if you would, for example, Google the videos of the sea angels hunting, you would see that they control each unique tentacle by which they try to catch the prey and also position this prey in some like locate this prey in some specific position and then to eat it because otherwise it would be very hard for them to consume the shell of the prey. And so later on, after more or less the same studies that Kandel did, so electrophysiology, some molecular identification, the neurons, these sea angels became a very important object for studying central pattern generators. I think you maybe heard about them during the lectures at GSN, for example. And now they are widely used all across the world, or some models that are very similar to them. And I just wanted to tell you this small story about the importance of this approach because, again, nobody expected them to be helpful in neuroscience. And now they became, after some time after the initial finding of these giant neurons in them and these central pattern generators, they became superstars. I was also very proud to see these animals because here you can see the biological station on the White Sea where they actually located and where practice of the physiologist of my faculty usually occurs. So just a small story to remember that selection of the model is very essential and that you should not uh, treat in a bad way the comparative approach also in neuroscience. And I think with this, I would actually finish my talk. Here is just another example from the same researcher. Here is our Russian Aplysia with the giant neurons and other mollusks that you can also use to study the same mechanisms. Also, a very interesting story, but maybe for another time. So, I give the word to Jenny, I think.
Yeah, that was cool. Russian Aplysia looks scary, as I would expect. Okay, guys. I think it was interesting to listen to about basic mechanisms, and we will see that in rodents, actually, it's really difficult to dissect how it exactly works, and we will see that soon. I think we can make 5-minute breaks if you want to digest some info, because we're studying learning and memory, and we know that if we don't have breaks to reflect on something, we can forget. So I don't want you forget about Aplysias. So let's make 5-minute break, drink some water, and then come back to discuss rodents. And thank you, Georgi. It was super cool. Actually, others, don't worry. I'm not expecting you to do as much work. So you will just present paper in short, like. But since Georgi like Aplysias, so yeah. See you in 5 minutes. Guys, can you see slides? Good. When I was in a break, I was also thinking that in general, all that simple organisms are really nice for computational neuroscientists as well, because like if you want to study how some simple system work, it's easier to model as well. So sometimes it's better to use Drosophilas or Aplysias or worms. But I know that like Drosophilas now just like mainstream in computational neuroscience. So people study how they made. Even I did internship with Drosophilas and tried to make them. Yeah, so it's kind of interesting. And also all that simple mechanisms, as Georgi said, they will kind of add up together in a more complex organisms with changes, but it's really conserved in evolution. And even like drug sensitization that we briefly discussed in habituation to different substances, it's all that synaptic plasticity kind of effects that could take place. So I think it's all interconnected. And sometimes it's really nice to see how it actually works in a simple organism, because sometimes it's really difficult to see the mechanism in complex big organism, because it's more difficult to dissect. A lot of stuff happens on the level of neural circuits and communications between circuits, and then game become really difficult. Yeah. So now we will discuss rodents. I hope it will not be boring, but I will try to give you main ideas why having like taking into account sensitization and habituation could be important. So let's go. Yeah. Okay. So I will just remind you. Of course, I hope you remember. Wait, something is yeah, like this. Um, Habituation. This picture really nicely represents habituation. It's a process that leads to decreased responses to a stimulus. So it was like a stimulus that you was reacting on, but then over time you understand that this stimulus is irrelevant, and then you can decrease your response over time. And it's like I really like a fairy tale about a boy who cried for wolf. It's basically a good example of habituation. It was a boy who was looking after his sheep, and he decided to like crack a joke and ask for help, like wolf, wolf, please come. And people came, tried to help him, but there were no wolves, and he did that several times. But then at some point, when real wolves came to eat his sheep, nobody came. Why? Because people habituated for his life. So it's like how habituation on behavioral level can work. But yeah, how to study habituation in rodents? So basically, there are several approaches, and we will discuss two main branches. One branch is special tasks. So when we work with animals, we usually first habituate them to the environment. For example, when I work with rats, I'm trying to habituate them to myself. So I'm doing handling. So rodents would know me. And also before starting some spatial task, I put them in the setup. So they would explore the setup and then habituate to the setup. So to reduce their anxiety level. And so what was shown that at the beginning of the trial or the session, rodents explore the environment, like in this case, open field, more extensively, and you can quantify that. There are different ways of quantification. For example, rodents prefer to stay closer to borders because it's for them a safe space. But then when they become more like familiar to the place, they cross the center, and here you can see how they're crossing the center, and all of that could be quantified. And then over time, when they learn the environment, their exploration level drops. So by that, we could say that, yeah, rodent is habituated, and basically it's kind of build a cognitive map. Cognitive map is a concept that was shown by O'Keefe and Nadel, and it's like really important in cognitive neuroscience, especially when you work with mammals. So I will cite that paper at the end of the slides, and you can take a look. Yeah, so basically once cognitive map is established, habituation completed. People call it like this, but nothing is super strict. Then habituation in spatial context like depends on hippocampus, right? We understood that when cognitive map is established, animal is kind of habituated, and habituation can happen within one session. For example, you have a session, and then at the beginning of the session, animal explores the environment more, and by the end of the session, exploration rate drops. And also intersession, when over several sessions, for example, you record the same animal over days, and in each day, exploration rate drops. So it also should be taken into consideration when we run behavioral experiments where rats are supposed to explore the space. Their behavioral is not constant. And here it's a theoretical example, it's not real data. It's what I found in one review, which is cited. It's actually a nice review, you can check, have a look. I also will cite it at the end. Here you can see the example how it would look like. For example, in a first plot, you can see that inter and intra-session habituation happens, like black bar showing exploration rate. Hello. Yeah. I need to take call. I'm having a class here and it's booked. Okay. Whatever. Yes, so black bars, it's exploration rate within first minute, and gray bars, it's like by the end of fifth minute. And here you can see that over days exploration rate drops. How to come back? Wait. Yep. So, and within one session, exploration rate drops. So here we can see inter and intra-session habituation. And here you can see different variation if only inter-session habituation happens. So within one session, nothing changed between first and fifth minute, and then over days it drops. Here you can see that between session, nothing changes, but within session, yes. And when no habituation, nothing changes. So it's like really visual explanation of intra and inter-session habituation. Well, and this spatial habituation mainly depends on hippocampus, because we discussed that cognitive map and everything, it's really hippocampus related. But then there are different approaches, and people show that actually when you impair hippocampus, that habituation drops. Okay, and then you can assume that hippocampus plays a main role in habituation, but it's not true, because there are different ways how habituation can happen, and those one was like only in context of space habituation, and also it could be interplayed with space learning. So when you learn the environment, your exploration then drops. How you can make sure that it's exactly habituation? Why you call it habituation? So a lot of questions appears. That's why research in aplysia way easier. You can look at molecular mechanisms and say like, yeah, this is habituation, this is sensitization. In rodents, it doesn't work like this. But there are different approaches, different from spatial approach. For example, there are studies that use startle response. We'll discuss that response later. It's basically when in response to some sudden stimuli, animal does like this. All muscles contract and animal is alert. So it's startle. And they show that actually in startle response, hippocampus mainly not involved. So it's another mechanisms. So we can see that hippocampus may be important in spatial tasks like open field, 8-arm maze, Morris water maze. And we will discuss those paradigms when we will discuss spatial memory. We will have it later in the course. But hippocampus is not really involved when we use reflex-like behaviors like startle response, leak suppression, or head shake response. Head shake response we will discuss soon. Yeah, so different approaches, different mechanisms, different pathways. For example, I found a nice paper that dissects those hippocampus-like habituation, hippocampus-dependent spatial habituation, and reflex-dependent habituation. And here they used head shake response. What is that? Head shake response. It's when you have a rodent, and when you apply a like mild air puff in the ear, it's irritating for them, and they kind of shake head like this. So it's like reflex. When somebody blows in your ear, you're like doing like this. And then over time, when you repeatedly do that, animal kind of get used to that because it's like mild stimulus. It's not aversive. It's not really strong air puff because otherwise it would not habituate. And what they showed here, it's ugly graphs because it's old paper, and old papers, it's always ugly graphs. Later in the course, we will discuss more modern topics with beautiful graphs, but for today, I'm sorry. So here you can see that what they did several types of lesions. They did sham lesion. It's when you do lesion in area that are not related to your area of interest. For example, you can do a small lesion in some cortex or some other area that are not important for your study. So it's like control. And neocortex and hippocampus session, it's where they did actually lesion in neocortex and in hippocampus. And here you can see that in general, response 5 minutes EC, it's intersession interval. So it's like you have 2 sessions. First, it's shown in a black, and second session shown in a white in all graphs. And 5 minutes here, it's like 5 minutes between those 2 sessions. Here it's 2 hours, 6 hours, 24 hours. But we, to be honest, don't care about that now. What we care about is that if these black squares or white squares, it's basically hippocampus lesion. But hippocampus is involved in a spatial like habituation, right? So we would expect, if it would be not the case, if we lesion hippocampus, habituation doesn't happen, but it happens. Yeah, you can see some kind of difference, but it's not very significant. So in papers, they basically said that, yeah, it's slightly increased responsiveness, but not so much. So basically, the main conclusion here is that all groups revealed comparable habituation in head shake response. So it means that in these types of tasks, hippocampus is not really involved, and spatial learning is not necessary to show habituation, right, in this case. Yeah, I think this is not really interesting. Maybe we could skip it, but let's try to dissect it. So here they also compared it with, I think it's not important. Let's move on. So another approach to study habituation, it's acoustic startle response. What is acoustic startle response? You can have animal in a startled chamber, for example, like in this image. And you have a grid that can detect the motion of the animal. And when it's startled, I told you that it's innate reflex characterized by rapid contraction of muscles. So when it's a sudden noise or something happens, animal contracts because it's like safety. And here you can see the nice example of kind of life example. Mouse is chilling, and then suddenly startle stimulus occurs, like cat meowing. And then you have a startle response, like rats contracted and try to kind of overcome the situations, like fight or fly, right? Something like this. And that's basically fly because you don't know how to fight. Yes, and here I found a nice schema of primary circuitry of this acoustic startle response. And you can see that here I cannot see hippocampus. So there is no hippocampus, but startle happens. So it's another pathway, another mechanism. And with this acoustic startle response, habituation also can happen. If you repeat several times this meowing to the rat, at some point in trial 40, it will be reduced response by this reflex, because at some point animal understands that there is no threat. There is nothing important in this stimulus, so habituation occurs. Yeah. And to sum up this habituation part, then we switch to sensitization. What's important, again, why we discussing this? First of all, as I told before, nowadays almost nobody studies habituations and sensitization like a main goal of the study. We should be aware about that because it happens even in other conditions. When you want to study special memory, when we want to study conditional responses, habituation can take place. So as we saw in spatial tasks, rats with lesions can respond differently. They can habituate worse, they can learn more worse, and it should be taken in consideration. Also, I found that environmental enrichment, sometimes it's used to kind of boost cognitive abilities of animals. And it was shown that environmental enrichment, for example, how you can do that, you can put in we call it like cognitive cages. You put in a rat cage a lot of toys, a lot of interesting stuff. I was even like in Moscow, I was building a cool big cage for my mice where they could like climb everywhere, run in a wheel, like different toys. I don't know. So when animals are kind of in a really enriched environment, it was shown that hippocampus thickness is increasing, and also dendritic arborization in granule cells increasing. So it can also boost your animal's abilities to habituate to novel setup to you and perform better in special tasks. So it's for special task. For conditioning that we will discuss next week, I didn't go to depth to that topic yet because I will postpone it for the next week. But also I found that it was shown that habituation may occur in classical and operant conditioning. So when you study these dependence between conditional stimulus, unconditional stimulus, and response, when you repeat it time over time, habituation can happen, and then it can influence results. So it should be taken in consideration. If you see some trend, it could not be the trend that you expect to see. And the last one, and it's important also when you work with animals, check how old your animals are. Because sometimes you train animals and then you postpone your experiments, like, okay, I will postpone them for some time because I need to do some surgery. Then your animals already like for half a year in vivarium. What can happen? If you use really young rodents, like it was shown that their level of habituation decreases. So when you try to habituate them to yourself, to train them and everything, their performance could be worse. Why? There are different factors. First of all, their exploratory activity was shown to be lower than in adults. And also because we know that in early development, neurotransmitters and receptors plays a bit different role than in mature brain. So we should take that in consideration. But usually it's not a big problem because it was shown for very, very young rodents, like 15 days old rats. So if you use, it's safe to use rats after third week after their birth, like 3 weeks, 3 weeks old rodents, fine. And the same can happen, the same differences, not the same, but difference you can see also in old rodents. Like, I don't know why, but I found this range 13, 24 months. I think like 13 is not so bad, like 1 year, it's not so bad. But when it's like more than 1 year, we know that cognitive decline happens, learning and memory become impaired, and of course, this influence habituation. So they will be less habituated, they will be less good at cognitive tasks. So it should be taken in consideration as well. And then we will switch to sensitization, but meanwhile, maybe you have some questions or you're bored or something. If nothing, I can continue with sensitization. Yep.
One question from me.
I
Sorry,
cannot
can you
hear
return
you.
to the previous slide? Wait,
I cannot hear you because I don't have my headphones. I'm sorry.
maybe this is. Can you hear me now?
Yes.
Yeah. So can you return to the previous slide for a second?
Yes, we can try. Yep.
So, yeah. So I was a little bit confused by these two facts. So we know that young rodents they have a less lower levels of exploratory behavior, but then we say that the habituation itself is worse in them. So I'm not sure how, like, I could not understand how is this connected. So am I understand correctly that the idea is that because the exploratory behavior is much, the activity of the exploratory activity is much lower, then there is no need to, in this strong habituation because they do not, so they do not need to adapt to the always, to this new environment because they actually do not explore it. Or like, can you explain me the link between these two
Okay.
facts? Yeah.
The logic is a bit vice versa. So for any habituation, like you want to habituate any animal because otherwise animal will not recognize you, will not recognize the environment and cause stress. Because novelty, it's not always fun. And when, for example, you handle threats, sometimes when they are not happy to see you, they are not happy to see you, and you should expose them to yourself over and over again. And with young rodents, since they're not exploring enough, so their exploratory activity decreased, it influences habituation. So it's kind of causality works in this direction. They explore less. That's why they cannot explore enough to habituate. They don't sample enough information in order to habituate. So they, over time, they will do the same, but they need way more time to habituate because they will need way more time to explore everything. And if it's spatial, like habituation, like an open field, they should spend more time to build that cognitive map. And then they can habituate. So you want them to habituate in order to be less stressed and like perform a task that you actually want them to perform. But for that, you should spend more time. It's easier to chill and wait until they become adults. And then you can do other stuff meanwhile. Yeah. Is it fine? Okay. So any other questions? Okay. Let's continue. Sensitization. Really nice example for me. I was sensitized, how to say it correctly, sensitized to water drops at some point. For example, normal person, you have irrelevant stimulus like water drops and it's dropping, and at some point you don't recognize that. You're like, yeah, it's dropping, whatever, you don't pay attention. But sometimes what happens is sensitization. You kind of, to the same stimulus, you overreact. And here I wanted to show that it can happen. So basically, sensitization is increased responsiveness to a stimulus. And it can be adaptive and maladaptive. What I described with water, it's maladaptive because it doesn't make any sense. Maybe it makes sense and could be adaptive for water waste, but it's already like, how to say, it's a joke. But like in a risky situation, you want to be over-sensitized to really dangerous stimuli, and sometimes it makes sense to overreact on them. Like, for example, I found a nice example with bees. When you hear a sound of bee again and again, then if once you got bitten, then when you hear that again and again, you will be overreacting and maybe run away, even though bees somewhere like far away from you. Why? Because you don't want an anaphylactic reaction and you want to be safe, right? And it could be maladaptive, for example, drug sensitization, since I'm pharmacist. I know about that a little bit. So when one uses amphetamines or opioids, it can happen. So in the first use, it's fine. It's how it's expected. But in the second use, it's like overreaction, and you can have overdose even with a small dose or with a dose with a normal range, or you can have a risk of addiction because that overreaction leads to some pleasant effects if it's drugs, and you kind of can be too happy and continue using them. So yeah, sensitization can be adaptive and maladaptive. Then how we can approach this question, how actually sensitization was studied in rodents. So in rodents, from what I found, maybe somebody of you knows a bit more, but yeah, sensitization usually studied in relation to stress, drugs, and like some drugs that connected to dopamine release, so like real drugs. And sensitization is bad and can be used to study in preclinical studies how drugs could affect animal behavior, and then you can relate it to humans and actually prognose and predict some side effects of certain drugs like amphetamines, cocaine, whatever. Then another branch is stress-induced sensitization. It's also important for those people who studies behavior in animals, because by some means we can cause stress in animals even without wanting that. And stress, it was shown that stress and constant stress, some pain can cause that overreaction in animals. And I mentioned pain already, but I say it again. There is a whole branch of research that studies like constant pain, chronic pain, and this chronic pain can also cause sensitization to not damaging stimuli. Because of that kind of level of stress and pain that persists, your organism is overreacting. You can even relate it to yourself. When you feel sick and just feel shitty, you can overreact on some stimuli. It happens. And let's discuss some behavioral paradigms. Again, acoustic startle response that we discussed before. And here another nice picture that can present that. For example, you have stimulus, like really loud meowing, for example, and you have a startle. But then over time, some background information stresses you a lot. Like here shown some blue monsters, but it could be some noise that we will discuss a bit later, or some light or some conditions that can cause stress to organism, and then on the same stimulus you overreact. And it was shown in a startle response. And we will discuss now briefly one paper that actually studied that sensitization in a startle by noise. So what they used the chamber that we discussed before and placed, I think they used rats. They placed the rat there and they presented them with different background noise, for example, from 60 decibel to 80 decibel. And it was like a constant background noise. It's not a noise pulse. And on top of that, they presented a tone that was 110 decibel. So it's quite loud for animals. And then they studied the startle response. So what they showed that, okay, like this, that 60 decibel noise, like in a figure 1 here, that 60 decibel noise was not stressful for animals. And you can see that in this case, habituation takes place. So here you can see in the x-axis, it's blocks of 10 tones. And on a y-axis, it's a mean startle amplitude. And at the beginning, when tone was presented on the top of that background noise, you can see that startle response is quite high. But over time, animal kind of habituated to that tone and does not respond anymore, because it's not like conditioning when you have a tone and then some food. Of course, you will take into consideration that tone and then you will use it for your advantage. But in this case, it doesn't make sense. So they habituate. But what happens if it's 80 decibel noise? So this level of noise is quite stressful for rodents, and you can see that over time, instead of habituation, sensitization happens. So rodents start to overreact, and they show these effects consistently over several days. So this sensitization on the top that happens because of that high level background noise persists over days. So sometimes habituation kind of can decline over time when animal get used to that, but in this case, no. But what they showed, once they stopped that background noise and dropped it till 60 again, this startle response disappeared. So here you can see 80, 80. What does it mean? It was like 80 decibel and continued with 80 decibel. So you can see that startle increased and was increased after. Then you have 60-60 when it was low level of noise and it didn't cause sensitization. And with 80-60 protocol at the beginning for 30 minutes, they had 80 that caused sensitization. And then they dropped it to 60, that background noise. And after 15 minutes, they saw that sensitization decreased. So sensitization in this case was reversible. When you remove that stressful background noise, animal came back to almost normal level, like 60-60 to control. And what we should keep in mind, it's like short summary about sensitization, why we should take into consideration that. When design your experiment, you should take sensitization into account to avoid misinterpreting increases in response. So sometimes you're like, yeah, learning doesn't work. And sometimes like, for example, in object recognition, there is a paradigm that we will discuss also later in seminars about special learning. There is a paradigm about object recognition. And if your animal, for some reason, explores object more or less, and it's not expected by you, you should like think maybe there is something stressful going on with your animal. Maybe there is some like bad conditions, like noise in your facility, or like in my facility once happened, my friend played violin with my rat, and my rat was constantly in the cage. And friend was playing violin for more than 1 hour, super loud. And when I came, my rat was like with fur like this, because of course it's super loud, and my animal was for sure under a huge amount of stress. So you should make sure that your animals are kept in a standardized conditions with standardized light level, because bright light also stressful for rodents, for example. Or when you restrain animal for a long time, you they really scared to be restrained physically. So interaction should be mild when you handle them. At the same time, they got stressed in big empty spaces. So you should provide animals in a cage with shelter so they could hide. And also sometimes people use like transparent red shelters. So animals cannot see through red, but we can. So for us, that kind of shelter is transparent, but for animals is not. And then we can provide animal with shelter, but we can see the animal from our side. Yes, and also if you use drugs and your studies are more medical, you should always double check the pharmacology of the drugs, and if there are some studies on sensitization, to avoid unexpected side effects. Yes, with this, I would like to finish my part and answer some questions if you have, and then I will tell what's coming next, and we call it for today. And you can also say me whether everything was like more or less digestible or it was too fast, too slow. Yep, you can.
Yeah. Just a few things. So
Mhm,
first of all, thank you. Actually, it was very nice, digestible. Yeah.
mhm,
I just wanted to add, not to ask, but to add something
mhm,
because I haven't mentioned it in my presentation, and I think you also kind of touched some of the things, but yeah, not in details. So I think it's important to point also that we know now that this sensitization and habituation are very basic learning mechanisms. And as you said, it could interfere with classical, like operational conditioning that we'll
mhm,
discuss later. And what also was noticed, and it's important also to remember when you conduct experiments, that these basic bricks of learning could interact with the higher order mechanisms. For example, in even in Aplysia, Kandel showed that, for example, you can use this mechanisms to influence the classical conditioning. So, for example, you can sensitize the animal, but the stimulus to which it's sensitized, it's actually the stimulus that is kind of not directly linked to the something relevant. So, like exactly in the classical conditioning experiment. So, for example, you can sensitize animal to some irrelevant, unnatural stimulus, and then link it to the some food stimulus.
mhm,
And the first part, the first thing, and also I think actually it's a, like half question, half node. So
mhm,
I know that in Aplysia they did it. So it's also important to remember that you can mix these sensitization and habitation. So for example, as well as with this noise, you see the
mhm.
sensitization to the loud noise, and then you can
Yeah.
decrease it by habitation with lower noise. So you need to remember that to interfere, that they could interfere, and if you
Mhm,
want to achieve some behavior, you can manipulate by varying different, I don't know, experiment conditions to receive the behavior that you desire
mhm.
to
So
desired behavior.
basically, yeah, it's true. You cannot habituate. Habituation, it's like uh, some kind of adaptation. We cannot call it adaptation because adaptation is umbrella term. But in general, you kind of adapt to some stimuli. And in order to adapt, that stimuli should not be overwhelming or too loud, too something, too bright. It should be in normal kind of level. And when it goes beyond, I think almost any stimulus you can use as a sensitization stimulus, for example, with a noise or like uh, with some light conditions, it's like light too bright, like I will be sensitized now. I will be super angry. But if it's like this, it's better, right? So I think in this case, you can kind of shift your paradigm a bit with playing around with intensity of some stimulus. But for me, the main difficulty is to dissect mechanisms, and you almost never can study it in how to say separately. You cannot dissect it easily. It's always something interferes with that, and vice versa. When you do other research like special blah 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 whatever conditioning, always that mechanisms come and play their role. So it's like that's why sometimes behavioral neuroscientists get frustrated because you cannot say for sure what's happening. Yes. Yep. You can ask.
So, as far as I remember, you said that the habituation was thought to be related with hippocampus initially, but then it was thought that it was not the case actually. So, why do you think they first think such a thing? Because of the synaptic plasticity or the capability of the
Yeah.
neurons over there, or?
You know, it's we cannot say that it's not the case. It's like different mechanisms because it depends what you call habituation. In neuroscience is always a problem of defining something. Habituation, it's kind of decreasing of response to something. And in terms of habituation to a space, you decrease your stress level to a novel environment, and then your exploration drops, but also some patterns of behavior, like animals become more brave, they're crossing a center in open field, because when they are stressed and just trying to explore, they try to keep closer to borders. So I didn't say that hippocampus does not play a role. It's just another way, and habituation can happen in a different systems. So when you have some reflex and habituation to that reflex, it's one system that bypasses hippocampus. And habituation to something that involved building cognitive map, it's hippocampus related. For example, habituation to a handler, habituation to a space.
A spatial, the spatial memory that you said.
Yes, yes.
Okay, thank you.
Sure. So any other questions? If not, let's discuss what's coming soon. Next time, I want to discuss associative learning, like another branch, and I want to discuss all conditioning, including fear conditioning, PTSD, whatever, that all under the same seminar. And I didn't pick one cool paper yet because there are many, and I want to pick something more or less fresh with nicer graphs and more recent applications. So I would do it today, tomorrow, and I need somebody who would like to read through. And also, don't be scared. For example, don't read a whole paper. Just try to dissect behavioral approach and understand why they use that behavioral approach. Some details regarding like scientific question is not super important for us, because people sometimes study some molecule or something that they describe forever, and then they switch to behavioral mechanism, behavioral setup. So we are interested here approaches to study learning and memory, not like exact scientific questions. So who would like to take conditioning? If nobody, I will be sad and have to read everything myself.
I can do it.
Okay, cool. I will send you a paper, let's say, until Friday evening. Is it fine for you?
Yeah, that's fine.
Cool. So then I will include all references in the slides, and maybe some main papers that I consider more interesting and worth to look at some point if you want. Not necessary. I will upload them to folder, because last time I just cited everything and then I was like, yeah, I'm tired. And also because it was history. Today is not history, so we can send you some papers and books. Maybe also Georgi have some PDFs of books that he recommended, so we can upload that in a Google Drive. With that, let's call it for today. I hope we all learned something today, and see you next Wednesday. Bye-bye.
Thank you.
See you.
Thank you so much.
Yeah. How to, yeah.